Good morning. In this presentation, we are going to be discussing important aspects of muscle physiology. And we are going to be focusing on skeletal muscle physiology. Briefly, we are going to compare the different types of muscle fibers, and we are going to mention the differences in the physiology between the skeletal muscle contraction and characteristics and the smooth muscle and cardiac muscle. We are going to dedicate more time when we move to the cardiovascular system to speak about the cardiac muscle. So in this part, the focus will be the skeletal muscle physiology. And we have a series of learning objectives that we are going to be covering. We're going to notice that most of them refer to the skeletal muscle. But again, we are going to do a comparison so we understand the relationship between structure and function in the different type of muscle fibers. Okay, when we say muscle fiber, it's the same as muscle cell. We're going to be describing their structure, the connective tissue layers that protect, surround different structures in the muscle. And then we're going to enter into some of the characteristics of the skeletal muscle contraction. We're going to explain what is a twitch, how the muscles generate force, how the muscles maintain a muscle tone, they generate tension. We're going to be referring to the sliding filament model or theory of muscle contraction, which explains great part of the skeletal muscle physiology and cardiac muscle physiology. Not everything is explained by this theory, but it's a very useful model to understand how muscles contract. We're going to be talking about muscle metabolism, mainly what are the sources of ATP and how these different sources are used. And we're going to be talking about the different types of muscle contraction, isotonic, isometric, and then we're going to also relate the muscle function to the nervous system function. It's impossible to understand one without the other because these muscles are effectors of the central nervous system. So we are going to be talking about the regulation of the muscle function, muscle contraction by the brain. We're going to mention the pathways that control the skeletal muscles. And we are going to be trying to understand what is a motor unit, the different types of motor units that exist in the muscles, how important they are for muscle function, and the order of recruitment of different motor units depending on what type of exercise we are doing, okay, what is the force that the muscles need to develop, for how long. And we are going to be talking about action potentials. The focus of this presentation is not the action potential, but we have to mention, remember I told you it's impossible to understand one without the other. It's an important relationship between the nervous system and the skeletal muscle. There is a phenomenon that is called excitation-contraction coupling. That is simply the conversion of the electrical signal from the brain, the action potential, into a mechanical phenomenon that is the muscle contraction. How this energy, electrical energy, the signal is converted into an action, okay, that is the muscle twitch or the muscle contraction. So we can start by having a comparison of the three types of muscle cells or muscle fibers. Here we have a table that describes skeletal, cardiac, and smooth muscle and compares their location, the type of neurological control, their main functions, the characteristics of the cells, the nucleus, and makes a reference to the cell-to-cell junctions that exist in some of these muscles. Well, skeletal muscle is attached to skeleton, to the bones, very important for locomotion, movement, for maintaining the body posture. Skeletal muscles are also in charge of producing heat. Shivering, for example, heat production is called thermogenesis when we are in a cold environment. and participates also in the control of the micturition, urination, and defecation. Okay, the voluntary control of these activities, and we contract the external sphincters in the urethra, anus, so we don't urinate or defecate unless we are in a convenient place. We refer sometimes to the skeletal muscle, or many times, as a voluntary muscle, and that is true. The skeletal muscle responds to the somatic nervous system. Okay, most of the functions that these muscles do are voluntary. However, there are some involuntary functions. Okay, it may work, for example, in performing the deep tendon reflexes, and also shivering is an involuntary response of the skeletal muscles. It's important to have that in mind. And the cells are typically long. They can be very long. For example, in the muscles of the legs, they extend from one end of the muscle to the other. They are cylindrical and they are striated. We are going to see that these striations respond to the presence of sarcomeres, these special structures, okay, that are present in skeletal muscle and cardiac muscle. The skeletal muscle cells are multinucleated. The nuclei are peripherally located towards the cell membrane, and there are no special junctions between the cells in the skeletal muscle. Okay, the cardiac muscle is in the heart, forming the walls of the heart, the contractile cells of the heart. Okay, this is an involuntary muscle. We don't decide anything about it. This is innervated by the autonomic nervous system, responds to acetylcholine, norepinephrine, and also epinephrine from the adrenal medulla. And it also has autorhythmic cells, pacemaker cells, that define the rhythm of the heart. Okay, the function is pumping the blood. This allows the heart to work as a pump, generating a pressure so the blood can circulate throughout the body. The cells in the heart are short, are branched cells, and are also striated. Okay, it also has sarcomeres like the skeletal muscle. Okay, the nucleus in the cardiac muscle cells is only one, is a single nucleus. There are some cells that may have two nuclei, but that is not very frequent, and they are located at the center of the cardiac muscle fibers. There are very important cell-to-cell junctions in the cardiac muscle, the intercalated discs that strengthen the unions between the cells. There are many desmosomes, and there are gap junctions that allow the exchange of ions, so the electrical activity, okay, can spread quickly among the entire heart, so the heart contracts as a unit, generating a more efficient pumping activity. Smooth muscle, finally, is present in the wall of the hollow organs, blood vessels, digestive tract, uterus, gallbladder, urinary bladder. Is involuntary. Okay, is innervated by the autonomic nervous system, responds to acetylcholine, norepinephrine. Okay, there are other neurotransmitters involved in the control of the smooth muscle that act more locally as paracrines. For example, nitric oxide, VIP. There are different others that will regulate locally the activity of these muscles. For example, when we refer to the enteric nervous system, okay, there are some neurotransmitters that control locally the activity of these smooth muscle fibers. And we have to mention that as part of this smooth muscle activity, there are also some sphincters present in the small intestine, large intestine, in the entire GI tract, esophagus, and also the internal urethral, internal anal sphincter that have their own specific physiological control. These smooth muscle cells, they have a characteristic that they contract if they are stretched. So they respond to stretching by increasing the intracellular calcium and contracting. And they also have autorhythmicity, some of them. For example, in the small intestine, we have the cells of Cajal, or Cajal properly. And this is because Dr. Ramon y Cajal, Spanish doctor that discovered these cells many years ago. Okay, these are also pacemakers that determine the rate of peristalsis and movements in the small intestine during different phases of digestion. The functions of this smooth muscle are many depending on what organ we are talking about. Peristaltism in the GI tract, bladder emptying, maintaining a blood vessel diameter or varying the blood vessel diameter, pupil diameter, accommodation of the lens. Also, there are some special cells that are called myoepithelial cells present in many glands, for example, mammary glands, endocrine glands, that allow the ejection of the breast milk or the secretion of exocrine secretions. And this is something that we have to fix here. See how we make the mistakes. And uterus, gallbladder, contraction of many different viscerae. The cells are very different to the skeletal or cardiac muscles. They are spindle-shaped. They don't have striations. They don't have these sarcomeres as the other two. The nucleus is single, centrally located, okay, and there are gap junctions in some of these muscles. We are going to see later the distinction between multi-unit and single-unit smooth muscle. And we are going to see the importance of these gap junctions in this smooth muscle. So let's talk about the relationship between the central nervous system and the skeletal muscle function. Okay, here we have two diagrams. And on the left side, we have how the brain from the primary motor cortex, okay, sends signals down, okay, to the brainstem, where we have the nuclei for different cranial nerves. Okay, we have one neuron making a connection in the brainstem, and then another neuron traveling directly to the skeletal muscle, for example, of the face. Let's say this is the facial nerve. So there is an upper motor neuron and a lower motor neuron. The upper motor neuron goes down directly from the primary motor cortex to a nucleus in the brainstem. Let's say this is the nucleus of the cranial nerve 7, the facial. So there we have another neuron that directly connects to the skeletal muscle in the neuromuscular junction. For the control of the rest of the body, we also have an upper motor neuron that goes down to the spinal cord. Okay, notice this, making a synapsis here in the anterior horn of the spinal cord. And from there, we have the lower motor neuron that makes a synapsis with the skeletal muscle fibers in the neuromuscular junction. So there are two neurons in the motor control, the upper motor neuron and the lower motor neuron. Now, these neurons, okay, are modulated, the function of these neurons are modulated by fibers from the basal ganglia, by the cerebellum, that allow the smoothness, okay, the precision, the intensity, okay, of the muscle movements. Okay, the cerebellum also is very helpful in coordinating, okay, to fine-tune the movements, doing complex sequences of muscle contractions, maintaining balance and posture. Okay, this is the motor, or these are the motor pathways, okay, of the our nervous system, how it connects with the skeletal muscles. And when we study this, we always, it's a good idea to study it in context. Okay, these movements will depend on the sensory information. Okay, for example, here we have the sensory endings on the skin, temperature, for example. And in blue, we have the afferent fibers going through the dorsal root of the spinal cord. There you have the neuronal body. And you have this axon that goes up, okay, enters the spinal cord. Then we have the second order neuron that goes to the thalamus. And from the thalamus, we have the third order neuron that goes to the primary sensory cortex. There are thousands of connections here, okay, within these two areas of the brain, the sensory and motor cortex. Okay, we here have the influence of the limbic system, emotions, memories, cerebellum. We have many connections here that will determine what is the response to that stimulus. Okay, if this is, for example, hot water, of course, the response is going to be a withdrawal of the hand from the stimulus. That response travels following the upper motor neuron, that synapses here in the anterior horn of the spinal cord. And from there, we have the second neuron, the lower motor neuron. That is the one that directly innervates the muscles that are going to produce, for example, the flexion of the biceps in order to withdraw the hand from that hot water. Okay, in that case, we also need the relaxation of the triceps, the antagonist. Okay, this connection here is more complex than what we have here. At the same time that we stimulate the agonists to contract, there is another branch that will inhibit the contraction of the antagonists, the triceps in this case, so we can do the movement as quick as possible. Okay, always when we study one concept, it's important to find the relationship between this concept and the previously learned concepts in physiology or in any other subject. Now let's go to the skeletal muscle. Here we have the layers. This is more anatomy, but it's important to also study the relationship between subjects that we are studying. Okay, muscles contain a very interesting structure. Okay, the muscle fibers, the muscle cell itself, these long fibers, contain special organelles that are the myofibrils, okay, that contain many different sarcomeres in series. Okay, here we have the mitochondria, many different myofibrils. Notice the sarcolemma, which is the cell membrane of the muscle fiber. There you have the nucleus. Okay, this is a cell. Each of these cells is going to be surrounded by a layer of connective tissue that is the endomysium. Notice that this supporting connective tissue contains blood vessels that are very necessary to supply to each of these fibers. Now, the fibers, the muscle fibers, are organized in fascicles. There is a bunch or a bundle of muscle cells that form a fascicle. The fascicles are surrounded by a layer of connective tissue. That is the perimysium, wrapping the bundle, this fascicle. Okay, the perimysium also contains blood vessels, and the entire muscle is surrounded by the epimysium. Okay, the epimysium continues and covers the tendon. Okay, above this epimysium, we have another layer of connective tissue that is called the deep fascia. Fascia is another term, very old term for connective tissue. Okay, above the deep fascia, we have a fat, which is known as the superficial fascia, is subcutaneous tissue, areolar adipose tissue, areolar tissue and adipose tissue. So these are the coverings, these are the structures that protect and allow the functioning of the skeletal muscle. Okay, then we have to go deeper in the structure of the skeletal muscle fiber. Here we have the structures we mentioned before. These are the myofibrils, which are organelles that are specifically present in skeletal muscle and cardiac muscle. Okay, the sarcolemma, as we said before, is the cell membrane. There are mitochondria. In the skeletal muscle, the cytoplasm is called sarcoplasm. And the endoplasmic reticulum is called sarcoplasmic reticulum. Sarco is a word from Greek that means flesh. So all the terminology that you find that contains sarco typically is related to muscles. Okay, maybe related to bone too, but we're gonna see that in more advanced studies. Now let's take a look at these myofibrils. Okay, here we have a representation of part of this myofibril. Notice that they contain the structural and functional unit or contractile unit of the muscle, which is the sarcomere. Okay, we are going to see the role of sarcomeres in muscle contraction. Okay, here we have a more detailed explanation about the structure of the sarcomere. and you can see different structures there. You see the composition, the different proteins. For example, actin, forming the thin filaments, titin, which are the elastic filaments, elastic proteins, like a spring that holds, these actin filaments. And then we have in the center the thick filaments that are made of the protein myosin. Here we have a more detailed view of the thick filament. Notice that it has these structures, that are that are called the myosin heads, that contain the actin binding sites. These are the structures that bind to actin when myosin and actin interact. It has also a place for the binding of ATP. Actin is different. Notice that is a double chain of protein. Contains other proteins that are called regulatory proteins. Troponin and tropomyosin are the regulatory proteins, as you can see there. Now, all this is a sarcomere from the Z line to the Z line. This has another protein there forming a line. Notice that there are areas where actin and myosin overlap, and areas where we have only actin. That's why when we see the muscle under the microscope, we see these striations, because there are lighter and darker areas. The areas where we have only actin are going to be lighter, and the areas where we have the thick filament, myosin, is going to be darker. That's how we have light, dark, light, dark, light, dark, forming striations. The lighter bands are called I bands, and the darker are called A bands. So notice that light has an I and dark has an A, so you can use that to remember. I for light, A for dark. This is a complex topic that sometimes is difficult to remember if you want to study this in much detail. But remember, we are studying physiology, not anatomy. So we are not going to make emphasis in remembering tiny anatomical or histological details. What is more important is to understand the role of actin and myosin in muscle contraction. For that, we need to explain a little bit a model, a theory of muscle contraction that is called the sliding filament model of muscle contraction. On the top part here, you can see the sarcomere, how it appears when it's relaxed. Notice the separation between the two Z lines, and compare that to the appearance of the sarcomere when the muscles contract, the contracted sarcomere. Notice that the two Z lines come closer together, and that happens because of the interaction between actin and myosin. These myosin heads that we showed you before will bind to actin, and with the help of ATP, these myosin heads are going to start pulling from the actin filaments until the two Z lines come or walk towards the M line in the center of the sarcomere. So notice that the length of the thin and thick filaments don't change. If you measure the distance between these and these and this and this is the same, this thick filament the same, it doesn't change in length. The only thing that decreases in length is the light band. The dark band, the A band, doesn't change. The light band is the only thing that will be smaller when you compare the relaxed and the contracted muscle. All this occurs after a signal from the motor neuron reaches the muscle. An action potential spreads through the sarcolemma, enters through the T-tubules in the muscle fiber, and that will produce the phenomenon that we know as excitation-contraction coupling. When this action potential spreads through the T-tubules, it will activate some calcium channels Okay, calcium channels will trigger the opening of the sarcoplasmic reticulum, leading to the release of calcium into the sarcoplasma. In the sarcoplasma, calcium will expose, okay, the binding sites of actin, okay, and that will allow the interaction between actin and myosin. And then with certain cycles, okay, of ATP, ADP use, the muscle contraction will occur. Okay, this phenomenon is very interesting to study if you are studying cellular biology, okay, or biochemistry. Okay, we are not going to enter into too much detail, just understand what is the role of calcium, what is the role of ATP in muscle contraction. Okay, we are gonna go back to the role of the lower motor neuron. Okay, we are gonna see what happens at the level of the neuromuscular junction, and then move to understand how the excitation and contraction coupling occurs. So here we have a representation of the synapses between the lower motor neuron, remember, from the anterior horn of the spinal cord, okay, and making a connection with a muscle fiber. There you have the axon terminal, okay, the synaptic end at the neuromuscular junction. We have a magnification of these. Okay, we have the synaptic end bulb. We have many vesicles there that contain acetylcholine. There you have the representation of the action potential reaching this area. When this happens, we are going to have some calcium channels here in the axonal end that will open. Calcium will enter, and that calcium will bind to these vesicles, triggering their docking, okay, in the cell membrane, and the fusion between the membrane of the vesicle and the membrane of the axon, leading to the release of acetylcholine into the synaptic space or synaptic cleft. Then these acetylcholine will bind to receptors on the sarcolemma of the skeletal muscle fiber, and the binding of acetylcholine, as you see here, okay, will lead to the opening of these sodium channels. Okay, sodium will enter. Remember, there is a greater concentration of sodium in the extracellular compartment than in the intracellular. So by this difference in concentration, sodium will enter and will trigger the depolarization of the sarcolemma cell membrane here. Okay, that will initiate an action potential in this muscle fiber. Okay, this area that is in contact with the motor neuron axon is called the motor end plate. Okay, that initiates the action potential, depolarizes the muscle fiber. Then this action potential will travel throughout the muscle fiber and will enter through the T-tubules. We also have to mention that this acetylcholine needs to be broken down. There is an enzyme here that is called acetylcholinesterase that breaks down acetylcholine, so we don't have a permanent contraction. We need to remove acetylcholine from there, deactivate it, break it down by the action of acetylcholinesterase, and then we are going to recycle. Okay, the choline part is going to be taken back. Okay, a process called reuptake of the neurotransmitter. This is a phenomenon that happens in many other neurotransmitters after they are released, reuptake of the neurotransmitters. You probably have heard about SSRI that are used to treat depression and other emotional problems. Okay, this is selective serotonin reuptake inhibitors. So SSRIs inhibit the reuptake of serotonin. So serotonin stays a longer time, making its action in neurons in the brain, so leading to a more a longer sense of calm and relaxation, and leading to sometimes permanent changes in the neurons if the treatment is done properly, if the patients take the medication for a long time. So here we have a closer view of the T tubules. You have there the sarcolemma. Okay, it looks like cheese. You have these little holes that communicate, okay, or allow the spread of the action potential down the T tubules, okay, that are in contact with this structure here, that is the sarcoplasmic reticulum. There is a complex system of calcium channels there, okay, that will spread the electrical activity until the calcium channels in the sarcoplasmic reticulum open, so calcium is released, and calcium starts activating the contractile mechanisms in the sarcomere. This process, this phenomenon, by which the electrical activity, okay, is converted into mechanical activity contraction, is called the excitation and contraction coupling, okay, that is essential for the muscle contraction. Okay, here we have more about the phenomena of muscle contraction and relaxation. Okay, here we have what is necessary for a muscle contraction to occur. We have here exactly the same thing that we saw before, the release of acetylcholine at the motor end plate, the opening of the sodium channels, the generation of an action potential, the depolarization that travels through the sarcolemma, enters through the T tubules that are connected to the sarcoplasmic reticulum. There you have the release of calcium. Okay, calcium will allow the exposure, okay, of the actin binding sites, so there can be an interaction between the thick filaments, actin, and the thin filament. The myosin, which is the thick filament, and actin, which is the thin filament. Notice the presence of AVP, adenosine diphosphate there, okay, that will lead to what we call the power stroke, which is a change in the configuration of this myosin head. Okay, it's gonna move in this direction, and since this is bound to actin, the actin filament will move in that direction. Okay, and leading to the muscle contraction, to the shortening of the muscle fiber. Now, when we want to relax the muscle, okay, we need to reverse all this process. Okay, we need to attach, it's not represented here, but we need to attach an ATP, adenosine triphosphate here. So the actin and myosin detach. We break this cross bridge that we formed here. So they detach from each other and the muscle can relax. Okay, another necessary step is that we have to remove the calcium from the sarcoplasm. So we need to take calcium back into the sarcoplasmic reticulum. And for this reuptake of calcium here, ATP is necessary too. Because now calcium is going from low concentration to high concentration inside the SR. Okay, so in a nutshell, calcium is necessary to expose the actin binding site and allow the interaction between actin and myosin, the cross-bridge formation. ATP, adenosine triphosphate, is necessary to detach, okay, to weaken the union between actin and myosin, so they can detach from each other. And ATP is also necessary to take or to remove calcium from the sarcoplasm and take it back to the sarcoplasmic reticulum. Now, after we mention what is the significance of calcium and ATP for muscle contraction and relaxation, we need to make a reference here to the different sources of ATP in the muscle. Okay, and it's a brief overview of the muscle metabolism. There are basically three sources of ATP. One is the stored ATP that we normally have in the muscles. Okay, this ATP has a very short duration, maybe for around 15 seconds. If you want to do a quick sprint or jump or something, we can use only this ATP and nothing else. Okay, this is stored in the molecule known as creatine phosphate. It's a creatine molecule with a phosphate attached to it. Okay, every time we use an ATP in the muscle, we are gonna obtain, we come back here, AVP and phosphate. Now this AVP will be released and we need to attach another phosphate to it to form ATP, adenosine triphosphate, and that is the role of this molecule, okay, creatine phosphate, okay, that will bind the phosphate from creatine phosphate to AVP. Okay, that reaction is catalyzed by the enzyme creatine kinase. Okay, as a result of this action, the creatine is going to be released and a new ATP molecule is going to be ready for muscle contraction. Okay, if we consume this ATP, okay, after 15 seconds, we continue, for example, running for around a minute or so, we are going to need to use another type of source of ATP, and that is glycolysis, anaerobic respiration. Okay, this anaerobic respiration uses glucose that may be obtained directly from the blood, okay, or as a result of the breakdown of glycogen in the skeletal muscle, or glycogen in the liver that provides glucose to the blood. In any case, glucose is going to be used to generate 2 molecules of ATP. Okay, and all this will depend on the presence of oxygen or no oxygen in the cell and the type of activity that we are doing. Okay, if there is no oxygen, glucose is going to be converted to pyruvate. Okay, pyruvate may enter the aerobic respiration in the mitochondria to produce a lot more of ATP, but if there is no oxygen, the anaerobic glycolysis will lead to the production of ATP and lactic acid. Okay, lactic acid typically travels to the blood, travels to the liver. In the liver, may be reconverted back to glucose. But if we are doing a very strenuous physical activity, lactic acid may accumulate in the muscle, leading to muscle fatigue. Okay, lactic acid will lower the pH inside the muscle fiber, so the contraction of the muscle is going to be almost impossible. Okay, but if there is oxygen, the pyruvate will enter the aerobic respiration or oxidative phosphorylation. The oxidative phosphorylation in the mitochondria is a very efficient process that takes glucose, pyruvic acid, or fatty acids or ketone bodies in order to produce tons of ATP, okay, and produce carbon dioxide and water. Carbon dioxide will be eliminated by the lungs. It's a very easy way of eliminating waste. And this process may also lead to heat production. Okay, for the production of heat instead of ATP, we need a process that is called decoupling. Okay, we decouple the oxidative phosphorylation, so mitochondria, instead of making ATP, okay, lead to heat production by the muscles. And this is a process that, in theory, may maintain the ATP production indefinitely. Okay, so could permit a very long exercise, for example, marathon runners, etc. So now we can take a look at the different types of muscle contraction. Okay, we can classify the muscle contractions as isotonic or isometric. Okay, the word iso means the same. Tonic is the same tone, so the tone of the muscle, the force, doesn't vary. Isometric refers to that there is no variation in the length of the muscle. Now, the variation in the length occurs here in the isotonic contractions, but there are 2 types. Okay, we may have a lengthening contraction, also known as eccentric, or a shortening contraction, which is known as concentric contraction. Okay, so during the isotonic, okay, the muscle length changes. During the isometric, the muscle length doesn't change. For example, when we are holding any object in the hand, the muscle is contracted, but the length doesn't change. During isometric exercises. Okay, we have exactly the same thing here. Okay, but now we can see the type of movement. For example, flexion of the biceps will lead to a shortening contraction. concentric contraction. Okay, when we extend the arm, we still have the biceps contracted, but it's lengthening. This elongation, this lengthening contraction is called eccentric contraction. And holding the object, holding the weight, okay, here the tension of the muscle will be equal to the weight, to the load. That will permit an isometric contraction. So after we mention the types of muscle contraction, okay, we have to make reference, okay, to another very important physiological aspect, that is the motor unit. Here we have a very important physiological and also anatomical concept, okay, the motor unit. What is a motor unit? Okay, a motor unit is a single motor neuron that you have here, the lower motor neuron, and all the fibers that are innervated by that motor neuron. Okay, on the left side, we have the representation of smooth muscle, spindle-shaped fibers. Okay, and we have these nerves of the autonomic nervous system that have these little vesicles there, these little dilations there that contain vesicles, okay, with acetylcholine. For example, acetylcholine can be released there, or norepinephrine, leading to the contraction, okay, of these, or relaxation, depending on what organs we are talking about, iris, for example, or GI tract, or sphincters, okay, contraction or relaxation of these fibers. We need to understand that there are different types of motor units and they have different functions. Okay, some motor units are small. They have very few muscle fibers and enabling the very fine motor control, for example, of the fingers, or for example, the facial muscles that allow facial expressions, or the tongue. And there are large motor units that contain many more fibers, so they will allow more gross movements. For example, the back muscles, the thigh muscles. And these motor units, okay, the different muscles will have a different combination, different composition in terms of motor units. And depending on the force that we are doing, depending on the exercise that we are doing, depending on the tension that the muscle needs to generate, we are going to recruit one type or two types or the three types of motor units. Let's take a look at how this happens and what is the order of recruitment. Okay, notice we are going to be talking later more about the different types of muscle fibers, classified depending on their metabolism. Okay, we have here a representation of tiny motor units. Okay, let's call them motor unit number 1. They contain more slow oxidative fibers, the type of muscle fiber that we call type 1, that mostly perform aerobic respiration. They depend on a constant supply of ATP. Notice that they are small motor units. Then we have in blue, okay, motor units that are larger, but not as large as the motor unit 3. Okay, these in blue contain a mixture of fast fibers, which are glycolytic, and oxidative fibers, which perform aerobic respiration. And then we have the motor units 3, which are very large, that contain mostly fast glycolytic fibers that perform mostly anaerobic respiration. So if we use them, we are going to produce tons of lactic acid. So depending on the muscle tension that we need to develop, notice this diagram here. Okay, we have on the y-axis, we have the tension that we need to develop, and on the x-axis, the motor units that we recruit to develop that tension. If we don't need to develop too much tension, for example, simply to maintain the muscle tone or the posture of the head on the neck, we are going to use only motor units 1, which are small, which use mostly aerobic respiration. They simply produce carbon dioxide and water, so there is no lactic acid accumulation. This may be used for a very long time. We can hold our head position for hours without feeling tiredness. But if we need to develop more tension, we are gonna recruit other group of motor units. Remember, these contain a combination of aerobic and anaerobic fibers. So we are gonna produce some lactic acid here. Okay, we can use this or develop this tension for certain time, okay, until too much lactic acid accumulates and we get muscle fatigue. But if we need to develop a great tension, then we need to use the third type of motor units, the ones that contain mostly glycolytic fibers that produce tons and tons of lactic acid, so we can use them for a very long time. Okay, the accumulation of acid is gonna lead to muscle fatigue very quickly. Okay, on the right side, we have something that represents the same idea, but representing the motor neurons and the size of the motor unit 1, 2, and 3. Okay, it's a representation of the same thing here. Any muscle will contain different combinations of type 1, type 2A, and type 2B motor skeletal muscle fibers, okay, that are innervated by, okay, each of them by a single neuron. Okay, notice that a single motor unit will innervate a single type of muscle fiber, okay, but muscles contain a combination of different types of motor units, 1, 2, and 3. Okay, the order is represented here in the same way, but simply with text. Small motor units, okay, are small. They innervate a few type 1 muscle fibers, while larger motor units contain more and larger cell bodies and innervate type 2 muscle fibers, type 2A, type 2B, which are the ones that contain a mixture of glycolytic and oxidative fibers or only glycolytic fibers. Below, you have some videos, okay, that I put there. Okay, the videos are organized depending on their duration. The first one, I think, lasts for 3 minutes, and another lasts for 6, no more than 10 minutes, and the last one is like 30 minutes. But with the first one is, okay, you simply get the idea of how this works. So the next topic that we are going to be talking about is the relationship between length and tension. Okay, and this has a great relationship with the sliding filament theory. Okay, to understand this, you can go back, okay, to this slide where we have the sliding filament. Okay, notice that there is a certain overlap between actin and myosin when the muscle is relaxed. Okay, and then when the muscle contraction occurs and there is interaction between actin and myosin, the myosin will pull from actin until the Z line comes closer, as you see here. Okay, we're going to be talking about this degree of overlap that exists when the muscle is at rest, okay, to understand the relationship between length and tension in the muscle. Okay, this diagram represents what is the ideal length of a sarcomere. The ideal length is the length that when the muscle is at rest, relaxed, will allow to produce a maximal tension. And this occurs when there is an overlap between the thick and thin filament that is between 80 and 120%. That is the ideal length that will produce or allow the maximal tension. If at rest, the overlap between actin and myosin, okay, is below 80% or is above 120%, notice that the maximal tension that the muscles can develop is very, very small, the percentage of the maximal tension. Okay, so simply if the actin and myosin are too close together, there is no room. Okay, there is no room. If the sarcomere is shortened beyond below the 80%, okay, there is a very reduced overlap. are extreme shortening, the thin filaments have nowhere to go. So there is no way that we can develop tension. Something similar happens if the sarcomeres are too stretched, very, very stretched beyond the ideal length. There is a reduced overlap. So it's very difficult to develop tension. Okay, and there may be even a moment when actin and myosin never found each other, never encounter, never interact. So it's impossible to develop any kind of tension. So next topic is the muscle twitch. Okay, what is a muscle twitch? It's a synonym of muscle contraction. Okay, a single contraction and relaxation is what we call a muscle twitch. Okay, notice this diagram here that is representing time on the x-axis and tension developed on the y-axis. After a single stimulus, there is going to be a period that we call the latent period. Okay, we need to remember to spread the stimulus across the sarcolemma, has to enter the T-tubules, has to activate calcium channels, has to release calcium from the sarcoplasmic reticulum. So the contraction will occur after a tiny latent period. Then we have the contraction, and then we have the relaxation that is normally longer and slower than the contraction period. We need to take the calcium away from the sarcoplasm, take it back to the sarcoplasmic reticulum. So it's gonna take longer. Okay, these are the different phases of the muscle twitch. Okay, the action potential is propagating, calcium is being released. There is the increased tension generation, formation of the cross bridges, shortening of the sarcomeres, and then calcium is removed, pumped back into the sarcoplasmic reticulum, so the muscle fiber returns to the resting state. So, talking about this muscle twitch, muscle contraction cycle, okay, we have to mention a phenomenon that is important in the skeletal muscle, okay, when we need to develop the maximal tension, and is the possibility of the summation of the muscle twitches or the muscle contractions. Okay, in the previous slide, we showed a single muscle twitch. If we electrically stimulate the muscle, it will contract and relax. But if we stimulate the muscle again here, okay, before the calcium is taken back to the sarcoplasmic reticulum, more calcium is going to be released, and we are going to have a stronger contraction that will be added here. Okay, imagine that we have a contraction here of this magnitude. If we stimulate the muscle again here, we are going to have another, and another, and another. Okay, so we can increase the tension that the muscle develops if we stimulate the muscle more frequently. So before the relaxation is completed, okay, we are going to stimulate again and again and again, and we are going to have what we call the summation, okay, of these phenomena. Okay, we are adding together, okay, the different processes of excitation, contraction, coupling. Okay, if we produce the stimulation very frequently, notice that we are not going to even see any sign of relaxation. Okay, there is no time. Mind that we go back to this diagram. Instead of doing the next stimulation here, we do the next stimulation here or here. Okay, we are not going to see any wave of relaxation. We're going to see this line moving on and on and on and on and on without any relaxation in between. And that is what we call tetanus. Okay, tetanus can be incomplete, okay, when the frequent signaling causes the muscle to reach a peak, but there is a slight relaxation, or can be complete when there is an extremely high frequency of stimulation, okay, that allow the contractions to continue. Of course, not forever. There is a moment when muscle fatigue occurs, okay, and even if we continue stimulating the muscle is not going to contract. So all those things are important. All those concepts are very important to understand how muscles contract and how we develop tension in the muscle. Okay, there are two ways: by stimulating the muscle more frequently or by recruiting more motor units in the muscle. But you know that there are skeletal muscles that are constantly working. Okay, we always have what we call the muscle tone. We can be sitting hours in front of a computer. Our neck doesn't get tired. Okay, some people have to be standing the entire day. And, well, of course, they get tired at the end of the day, but they do this activity for some hours. Okay, we need to understand the concept of muscle tone. Okay, skeletal muscles will be rarely completely relaxed. Okay, they always have a small degree of contraction, even when we are not moving. Okay, this state of permanent contraction, okay, small degree of contraction, is called the muscle tone, and that is vital to maintain our posture, stability of the joints, to have the muscles ready to initiate an activity if it's necessary. And this is a very complex phenomenon. There is an interplay between the nervous system, the cerebellum, the basal ganglia, skeletal muscles, nerves, and it's a complex phenomenon in which the brain decides, okay, which motor units are going to be active in a single time. Let's say you have in a muscle 100 motor units. We are going to use 20 of them during 10 minutes. Then we are going to let them relax and use other 20. Then let those relax and use other 20. And we alternate in a cyclical pattern the number of motor units and the individual motor units that we are using. So some motor units can recover while other motor units are actively producing tension. So the muscle never gets completely fatigued, and we can maintain a constant muscle readiness, and we can maintain our body posture. We can maintain our muscle activity. It's a very interesting phenomenon. And we are gonna finalize this part here with the presentation of this table. Okay, that is gonna be very useful for you to study, for you to prepare. We have here the different types of muscle fibers that we mentioned before. And remember, we have the fast glycolytic fibers, which are the ones that perform anaerobic respiration. These are called type 2X or type 2B sometimes, or fast twitch. Okay, then we have the fast oxidative. That is that they have in common the fast of these and the oxidative of these ones here. These are the type 2A that also produce fast twitches, not as fast as these ones. And then we have the type 1 or slow twitch fibers, which are slow fibers, oxidative fibers. Now, what are the differences? Remember, one of the important things is the type of metabolism they have. The fast glycolytic are glycolytic fibers. They have mostly anaerobic metabolism, okay, compared to the slow oxidative that have aerobic metabolism. And there is a combination in the fast oxidative, type 2A. Now, to perform aerobic metabolism, it's important to have a high number of mitochondria. Okay, these fast glycolytic have very low mitochondria. And there is a mixture in the ones in the center, the type 2A. The same thing happens with the resistance to fatigue. The fast glycolytic are not resistant, they very easily fatigue. The slow oxidative are very resistant to fatigue. And the ones in the center, that is a combination, remember. Now, there is an enzyme, the ATPase, okay, that will have a very high, very fast activity in the fast fibers. It's going to be simply high in the fast oxidative, and it's going to be low, the slowest, in the slow oxidative. So as a result of that, the slow oxidative fibers are going to be very efficient. Okay, they are very resistant, very efficient, while the fast glycolytic fibers are going to be or have a low efficiency because they have a low resistance to fatigue. Now, there is another thing that is different in these fibers: the amount of myoglobin and the amount of glycogen. Okay, a way to remember this is myoglobin is a molecule that is very similar to hemoglobin, stores oxygen, and glycogen is a molecule that stores glucose. Okay, so which are the fibers that need more oxygen? Well, the ones that have more mitochondria. So the slow oxidative fibers are the ones that are going to be or have the highest number of mitochondria. And this is what gives the red color to the fibers. Okay, another name for them is red fibers. While glycogen is what supplies the glucose for the glycolysis. So the fast glycolytic fibers will have a very high content of glycogen. Not too many mitochondria, not too much myoglobin, not too many mitochondria. So they look pale. Okay, when you go to the supermarket and you see the meat, okay, this very high quality meat, for example, filet mignon, okay, will have a very high number of slow oxidative fibers, which are soft because of the number of mitochondria and the very low content of glycogen. They are very red, beautiful color. First quality meat type 1, most expensive, red because of the myoglobin. Now, if you buy a meat that has more of these fast glycolytic fibers, it's not going to be as beautiful, it's not going to be as red, it's going to be harder. Okay, you need a pressure cooker for it. Okay, and the color is because they don't have too much myoglobin. They are more pale because they have more glycogen. Okay, relate glycogen to sugar. Sugar is white, pale, while myoglobin is related to hemoglobin, to blood, red. Those are some mental helps that we can use to remember these things and make sure we remember them forever. Okay, those are the types of muscle fibers. Okay, you always relate these concepts to what we saw here about the type of fibers that are contained in the motor unit 1, small, contain mostly slow oxidative. That's why we can use them for a very long time and to develop only activities that require small or not too much tension. Mixture of fibers and only glycolytic fibers. Okay, greatest tension, greatest power, but not very efficient, okay, because they easily fatigue. More efficient are the ones that we use all the time or most of the time. Okay, so we are going to make a brief reference to smooth muscle and cardiac muscle. Remember, the focus of this presentation is skeletal muscle. The important thing to remember about smooth muscle is that these fibers are spindle-shaped, they have a single nucleus, and these are fibers that can undergo hyperplasia. In the case of the skeletal muscle, something that we didn't mention before, skeletal muscle fibers don't divide. Okay, they can undergo hypertrophy, they can grow larger, and we don't develop more number of muscle fibers, skeletal. In the case of smooth muscle, yes, they divide, they can undergo hyperplasia, and they are not striated, they contain actin and myosin, but not sarcomeres. Okay, the contraction is a little bit different. In the case of the smooth muscle contraction, it will use calcium from the extracellular fluid. They also have smooth calcium inside, but they also use calcium from the extracellular compartment. That is something that doesn't happen in the skeletal muscle. Remember, skeletal muscle fibers are surrounded by connective tissue, so they are unable to get calcium from the outside. Okay, there is also a phenomenon of cross-bridge formation between actin and myosin, but a little bit different from the skeletal muscle. We don't have this sliding filament or sarcomere mechanism. Now, another difference between smooth muscle and skeletal is that smooth muscle can be stimulated by itself. Pacemaker or pace setter cells can be stimulated by the autonomic nervous system, hormones. They may have spontaneous activity or by stretching. Okay? And the smooth muscle also has the capacity of forming what we call latch bridges, okay, that enable long-term contraction without using any ATP. That is important, for example, in the case of sphincters that need to be closed the entire day. If we need to have a sphincter contracted the entire day and we would require ATP for that, okay, that would consume the entire energy, the entire nutrients of the body. So formation of latch bridges will allow the contraction without using ATP. Now here we have a couple of details, okay, a couple of different types of muscle, smooth muscle units. We have one that we call the single unit. Okay, these smooth muscle fibers contain gap junctions. So these gap junctions allow the exchange of ions. So the action potentials will spread across the entire group of smooth muscle cells. So they can act as a unit. They can have a synchronized contraction. This is found in visceral muscle, okay, for example, in the gastrointestinal tract. On the other hand, we have the multi-unit smooth muscle. They don't have these gap junctions, so they don't share ions. The contraction doesn't spread from one cell to the next. So we may have a more precise location of the activity. We can contract some fibers and not other fibers. Okay, these are present in the airways, in the large arteries, also in the erector pili muscles in the hairs. We have also in the pupil, in the muscles that produce accommodation or change the shape of the lens. Okay, we have here another details about the smooth muscle fibers to understand how the contraction occurs. Okay, we have, instead of sarcomeres, we have structures that are called dense bodies. Okay, and we have intermediate filaments of actin and myosin that are forming like a network throughout the sarcoplasm. Okay, these dense bodies are very similar to the Z lines of the sarcomere, the proteins, but they don't form sarcomeres. So when the smooth muscle cell contracts, okay, it becomes almost like a squared structure, like a post stamp. Okay, so it's a different type of contraction if we compare this to the skeletal muscle. And finally, we have to mention the characteristics of the cardiac muscle. We already saw the table before, but here we can see the characteristics. Okay, the cardiomyocytes or cardiac muscle cells, they are shorter, they have smaller diameters, they have striations because they have sarcomere. Okay, and they also have T-tubules that allow the electrical impulses to enter. Okay, they have a single nucleus. They may have more than one. Okay, and they have some differences. Okay, between with the skeletal muscle, not only the shape, not only the morphology. Okay, one great difference is the presence of intercalated discs. Okay, there are junctions between adjacent cells. They contain desmosomes, they contain tight junctions, and they contain many gap junctions that allow the synchronized activity, okay, of these cardiac muscle fibers. So the heart can produce a very efficient contraction. Okay, this is very important for understanding how the heart works as a pump and understanding the efficiency of the heart in developing the blood pressure that is necessary to send the blood to different organs. Now, another difference is that cardiac muscle cells also get calcium from the extracellular compartment. That this is something that doesn't happen in the skeletal muscle contraction. When we move to the study of the cardiac physiology, we are going to see something about the action potentials and about the muscle twitch in the cardiac muscle. Okay, that is important for the cardiac muscle contraction, the efficiency of the heart as a pump. Okay, there is a great, very important difference in the action potential that occurs in the skeletal muscle fiber and that occurs in the cardiac muscle fiber. But we are going to leave that information for later. I don't want to give you all the secrets now. So this is what we had for today. Okay, this is what we had about the muscles. And hope you learn a lot, hope you continue studying a lot and can apply all these in the future.